Welcome to the William N. Pennington Engineering Building.
This building is the newest addition to the College of Engineering and is home to 40 laboratories, a Class 100 cleanroom, a large computer lab, 150 graduate workstations, and a 200-student classroom.
So the laboratory also hosts several cubicles for the laboratory staff to work in when they're not in the lab. So as you see, they have computers at their workstations. So staff can research topics, they can work on assignments, or they can analyze results when they're not doing things in the lab. So a prime example of this is our very own Bella Doe. She's a graduate student here at the University of Nevada, Reno. And another graduate student we have here is AJ Bolt, graduate student for chemical engineering. So the lab we're going to be touring today is the lab of Dr. Mariam Sarmazdeh. She joined the University of Nevada, Reno in July 2019, and her lab focuses mainly on biomolecular engineering and synthetic biology to develop novel therapeutic tools. So let's head on in.
The research in the Sarmazdeh lab is focused on engineering biomolecules, especially proteins, using directed evolution and rational design with applications in biomedicine, drug delivery, biomaterial, and bioenergy. Proteins are workhorse molecules in nature, and our lab is interested in expanding their function using state-of-the-art protein engineering techniques.
So this is the thermal cycler, which is used to amplify DNA in the polymerase chain reaction, or PCR for short. So the way it works is the samples are heated inside of this thermal block using pre-programmed steps from the machine. Gel electrophoresis is a technique used to separate DNA according to its fragment size. So first a gel is created using agarose and 1x TAE buffer. The solution is heated until the agarose dissolves, and then ethidium bromide is added to serve as fluorescent tag. This solution is then poured into a gel cast, and the gel cast has a comb within, and this comb creates little wells within the gel. The DNA is then aliquoted into these individual wells, and then the entire gel is placed within this cassette, where an electric current is run through it. Once this process has completed, the gel is then imaged on a plate and within the gel imager. So this machine is a larger centrifuge, which is used for greater volumes of solution. So there are several different fridges and freezers in this lab, but this freezer is particularly important as it contains all of the protein samples that they use for testing. So the fridge is kept at negative 20 degrees Celsius in order to preserve them, and all of the master mixes, loading dyes, and buffer solutions are within this freezer.
The FPLC performs fast protein liquid chromatography, which purifies the target protein from the sample. Size exclusion chromatography, or SEC, separates by size. Ion exchange chromatography, or IEC, separates by charge. And metal ion affinity chromatography, or IMAC, separates by affinity for different metals. The flow cytometer counts tens of thousands of cells, one at a time, and can distinguish between them by different physical or chemical properties. For instance, our cells are attached to fluorescent antibodies that can be detected by the flow cytometer. The plate reader analyzes and compares up to 96 samples at once using a 96-well plate through fluorescence, absorbance, or other methods. One example is enzyme activity, which is detected via fluorescence when a fluorescent substrate reacts with the enzyme. The nanodrop can use just 1 microliter of sample to detect its concentration, purity, and other characteristics. We use it for DNA, proteins, cells, and other molecules. Next, we will be looking at a couple of machines in the cell culture room. The Biosafety Cabinet, or BSC, provides an ultra-clean working environment when working with precious cells or samples. It uses filtered air and UV light to keep the workspace clean, which is necessary for a mammalian or human cell cultures to thrive. The CO2 incubator mimics conditions inside the human body, like temperature, pH, and humidity, for growing mammalian or human cells. CO2 is pumped into the incubator, which reacts with water to form a carbonic acid and bicarbonate buffer that maintains a neutral pH.
The exciting experiments that take place in this lab are aided by all of the machines you just saw. The lab staff use these machines every single day. And with the help and dedication of every researcher, the research and results are made possible.